So at this time, I would like to introduce Stephanie Strathdee, Associate Dean of Global Health Sciences at the Department of Medicine, University of San Diego, and she's co-director of the Center of Innovative Phage Application and Therapeutics. She and her husband, Tom Patterson, who's professor of psychiatry at UC San Diego, co-wrote a book, The Perfect Predator, describing an unbelievable struggle to save her husband's life from a totally resistant bacteria, and how this journey brought together 3 national research centers and resurrected a forgotten therapy that using phages to kill deadly bacteria, this can be extremely effective, even though it was largely discarded by Western medicine.
Thanks very much, Kevin. Can everybody hear me? It's a pleasure to be here. I'm actually joined by my husband, Tom, who you'll hear from at the end of the presentation. So, spoiler alert, he lived and he's alive to tell about it. So our story began when we were traveling in Egypt in 2015. This was on Tom's bucket list, and we traveled there right after a terrorist attack in Sharm el-Sheikh, and we were nervous that we were going to have some turmoil, but we didn't know that the danger was going to come from within, because Tom was eating this wonderful seafood paella dinner on top of the cruise ship with me on our last night in Luxor at the Valley of the Kings, and he came down with what I thought was food poisoning. He was vomiting uncontrollably for about 24 hours, and I was insisted on bringing a doctor to the ship who gave him intravenous antibiotics and some fluids and said he'll be right as rain in a few hours, and he wasn't. He actually got worse. And this photo here on the left is Tom in a CT clinic. Luckily, they didn't have a hospital in Luxor, but they did have a clinic where they were able to image his abdomen, and they didn't like what they saw. They didn't exactly tell us what was going on, but luckily we had travel insurance, $38 paid for ultimately 7 ambulances and 2 Learjets to get him home. But he was too weak to get flown back to our home in San Diego, so they sent him to Frankfurt initially. So the photo on the right-hand side is us in the ICU at the Uniclinic in Frankfurt, where you see that I have a mask on because they put us in isolation. Initially, I thought this was just a precaution. They said, you know, you're coming from Egypt and there's a possibility that he could have some kind of infection. But honestly, I really didn't take that very seriously. And they found, after they repeated the CT scan, a giant abscess in his abdomen the size of a small football. And they said that they believed it was caused by a gallstone because they removed a gallstone from his biliary duct. But they also showed me this fluid that was dark and murky and putrid. And they said this abscess had just formed. This fluid would be clear, but it's obviously very murky and there's something growing in it and we have to culture it, and let's hope that it's a garden variety organism that will respond to antibiotics. So I still wasn't terribly worried at this point, but then when they diagnosed him along the way here with pancreatitis, which was from this gallstone, and he started to develop some serious complications, so we called his daughters and they came to Frankfurt and we hoped that he would pull through, and he did. But a couple of days later they got the culture results back, and this is just a montage of some of the shots from our journey. I posted these on Facebook thinking that he was going to be out of the hospital and we'd be laughing about this in a few days, but little did I know that this was the beginning of a very long journey. So what they found in Frankfurt after they cultured this organism from his cyst in his abdomen was acinetobacter baumannii. Its nickname is Iraqibacter because so many veterans come back from the Middle East with this pathogen. And it ultimately became the #1 deadliest superbug according to the World Health Organization just a few months earlier. And I was shocked by this because I have a degree in infectious disease epidemiology, but I'm not a medical doctor. And this organism was one that we used to plate on our Petri dishes back in the 1980s when I was a microbiology student at the University of Toronto. It was considered to be a really wimpy pathogen back then. We only needed gloves and a lab coat, and we were undergrads. But over the last few decades, this organism has acquired superpowers. It's essentially a bacterial kleptomaniac that steals antibiotic resistance genes from other organisms, and that's why it's earned its place on the superbug list. And when an organism like this is detected, the first thing that doctors do is develop an antibiogram, which essentially is the antibiotic resistance profile. And this is what they showed me while he was in the ICU in Frankfurt. It's all in German, of course, but you don't have to be a rocket scientist to see that all of these different antibiotics that are listed in this chart on the right-hand side of this slide are resistant. So essentially all of these R's mean that this organism is resistant to all of those, and it was only sensitive to colistin and a couple of other ones, minocycline. So they immediately put him on these antibiotics, but colistin, for those of you who are in the medical profession, you'll know that it is one of the last resort antibiotics, and it's reserved for extreme cases because it's extremely toxic to the kidneys and other organs. So they stabilized him and they medevaced him home to San Diego, where he was now being treated by our colleagues at the University of California, San Diego, where Tom and I are both faculty. And so I was really relieved, thinking, okay, we're back home now. We have some of the top infectious disease doctors around the world caring for him, and now he'll be fine. But he wasn't. In fact, by the time he got medevaced home to San Diego, this organism was resistant to all remaining antibiotics, even colistin. And the doctors were shocked because this resistance to colistin had just recently been reported a few months earlier. It was identified in China, the MCR1 gene that confers resistance to colistin. But by the time we looked, it was in 30 countries, and so therefore it wasn't a surprise that Tom had it. But that's how quickly organisms like this can spread resistance, and especially if we're not engaging in global surveillance to monitor it. So this photo was taken actually after the doctors told me that Tom wasn't going to make it. He had been in the hospital now several months. This photo was taken in mid-February, and I was really beside myself. I asked Tom if he wanted to live, and if he did, I said, please squeeze my hand and I'll leave no stone unturned. And I waited, and he squeezed my hand within about a minute, and he squeezed it really hard. And at the end, he'll tell you what he was thinking and hearing when I said that. But at the time, I didn't know, because he was on a respirator because his lungs were failing. He was on 3 different heart medications or pressors to keep his heart pumping, and his kidneys were hanging on by a thread. And when he squeezed my hand, I decided I would take matters into my own hands. Even though I'm not a medical professional, I do know how to do research. And I went home and I hit the internet, and I found this paper which identified a number of different alternative treatments for multi-drug resistant acinetobacter baumannii infections. And one caught my eye, which is phage therapy, or bacteriophage therapy. Now, because I had this ancient degree in microbiology, I knew what phages were, but just in case you don't, phages are viruses that have naturally evolved to attack bacteria. This is a scanning electron micrograph showing phages in the green attacking a bacterial cell in the orange, and they are 100 times smaller than bacteria, so you can't see them with a light microscope, but they almost look like alien spiders from outer space, and they're attacking this bacterial cell, and they take over the machinery of the bacterial cell and turn it into a phage manufacturing plant. And so I thought, wow, this is a great idea, because when it does that, it ushers a kill signal, and these baby phages burst out of the bacterial cell and then go on to attack new bacteria, and so on, until the bacteria that they match to are all dead. And then they are naturally excreted by the liver and the spleen of the body, so they don't harm the friendly bacteria in our microbiome. Well, I thought this was a great idea, and I turned to Tom's doctors, who were our colleagues, and I said, could we use this phage therapy to treat Tom? And what I didn't realize at first is that phage therapy is experimental treatment in North America and most of Western Europe, even though it was discovered 100 years ago by a French-Canadian who went on to use it in animals and in people quite successfully. And we can get into this discussion later, but phage therapy had a heyday in the 1920s and 30s, but when penicillin came on the scene, amidst other factors, phage therapy was forgotten in the West, and it was continued only in parts of Eastern Europe and the former Soviet Union, where antibiotics were in short supply. So basically, this meant that we had to get approval from the Food and Drug Administration to administer an experimental treatment like this. But first, we had to find phages that would match Tom's bacterium, because phages are very specific. They only kill the bacteria that they are matched to, so it's almost like a lock and key. So I took this photo of Tom where he has this t-shirt, "I survived Iraqibacter," draped over him by one of our students. And he wasn't surviving very much at all at this point. And I sent this photo to total strangers around the world, begging them to help. And I did this because I made a list of researchers that were studying acinetobacter, baumannii, and phage. And I did this using PubMed, which is PUBMED. The National Library of Medicine has made this free search engine available. You can punch in any keywords and find scientific literature. So that's how I found researchers working on this. I restricted my search to North America, thinking that we weren't going to have very much time to save Tom. And a researcher from Texas A&M, pictured on the left, that's Dr. Rai Young. He basically has been a phage researcher for decades, was close to retirement, was really touched by my email, and he said, "If you send me Tom's bacterial isolate, I'll turn my lab into a command center and try to see if we can find some phages that will match." So this is a picture of his lab team, including Adriana Carolina, who was a PhD student who slept in the laboratory for weeks trying to find phages to match Tom's. And she did. They found 4 phages that matched his isolate, which is quite amazing because they only had a few phages that they'd identified over the last 7 years. And when you go on a phage hunt for these perfect predators that attack bacteria, you have to go to places where you're going to find a lot of bacteria, because that's where you are most likely to find what's preying upon them. So if you're looking for lions, you go to where you see zebras and gazelles. But if you're looking for phage, you go to sewage, because there's a lot of bacteria in sewage. So literally, they combed through sewage samples and samples from barnyard waste, etc., and found 4 phages that matched. So now we were very excited, and my colleague turned to the Food and Drug Administration and said, "We have these phages that we want to try to save this man's life." And she said, "Well, there's a couple of other teams that are working on this in the U.S. that you might not be aware of." And he said, "Well, Stephanie's done a very thorough search. I don't think she's missed any." And she said, "Well, I don't think she knows about this guy, who's a dead ringer for Tom Cruise, but it's not Tom Cruise. This is Lieutenant Commander Theron Hamilton from the Navy Medical Research Center in Frederick, Maryland, who had a phage library that they had been developing, had never been used on humans. And he said, we'll look as well and see if we have any phages in our library that will match. So he turned to his colleague, Dr. Biswajit Biswas, who is affectionately known as the phage whisperer, and they found 4 phages that match. So this could be better news. We have now 8 phages, and you want to have several phages in a cocktail because the bacteria and the phage have co-evolved for millennia. And if you only have one phage, then the bacteria are given the upper hand to develop resistance very quickly. Ideally, you want different phages that attack different receptors of the bacteria. So we didn't have the luxury of time to know how similar or different these phages were, but we hoped that we had phages that would be able to keep the bacteria in check. And so we took this to our investigational pharmacy after we got approval from the FDA. This is the Phage Preparation that was prepared, and they were very meticulous because they'd only dealt with experimental drugs, and this is the first time that the drug is alive. This is what Tom looked like the day before phage therapy began. As you can see, it just looked terrible. He had 5 different catheters in his abdomen trying to drain this infected fluid out of this abscess. He was on a feeding tube, the ventilator, 3 different pressors that I mentioned, but now his kidneys were failing. So literally he was within hours of dying, I was told. And it was the scariest time of my life. We administered the phage first into the catheters in his abdomen, thinking that this was the closest to the infection. But previously, one of these internal drains had slipped and dumped all that infected fluid into his body. So he was fully colonized with this superbug. So some of the experts we consulted with told us, look, you need to treat him intravenously because if there's a hidden reservoir that these phages can't reach, the bacteria will become resistant very quickly. So since he was stable after administering the phage, 2 days later we injected the phage into his veins, 1 billion phages per dose, and we just prayed that he wouldn't undergo septic shock. This was actually the first time that phage therapy had been used intravenously in North America to treat a systemic superbug infection. And 3 days later, believe it or not, Tom woke up from his coma and lifted his head off the pillow and kissed his daughter's hand, who was standing in front of him. And everybody in the ICU freaked out. Nobody expected him to live. It was just a really remarkable turnaround. Because he was an N of 1, we can't prove that for sure that the phage were the reason, but all of the antibiotics that he was receiving were not working until the phage therapy began. And in fact, one of the phages ended up being synergistic with one of the antibiotics, and we can talk about that later if you're interested. So Tom began a very long recovery. He'd lost 100 pounds, all of his muscle mass, so he was in the hospital a total of 9 months. We believe that if he'd had phage therapy very early in the course of his illness, he could have left the hospital by walking out instead of leaving in a wheelchair. You'll see him in the wheelchair here. So he left the hospital August 12th of 2016, and it was really the beginning of what was to become a watershed moment in the history of phage therapy. This slide actually is a photograph and a scanning electron micrograph that was given to us by the Department of Homeland Security. So I showed you a phages attacking a bacterium before, but this is Tom's bacterium in blue, so that's a stain, being attacked by all of these hundreds of baby phages that are stained in green. Tom's case was presented at the 100th anniversary of the discovery of bacteriophage in April of 2017, and from that moment on, it went viral in a good way. We were profiled in Time magazine, the Huffington Post, the Washington Post, journals like JAMA, the Lancet, etc., and it still actually is going on to this day, even though it's more than 3 years past. And that's because we're really on the cusp of a post-antibiotic era, where by the year 2050, 1 person every 3 seconds could be dying from a superbug infection. So the story doesn't end there. In fact, we've treated many other people at UC San Diego, a total of 11 people now, with different kinds of infections, different kinds of underlying health conditions, and most of the cases have been treated successfully. The second patient, John, is shown here. He received a lung transplant and had a Pseudomonas infection that he couldn't clear, and the phage cleared him within a matter of weeks. This patient, patient 6, is Joel. These photos are shared with permission. Joel had a left ventricular assist device, which essentially is a device that pumps your heart for you after your heart has failed. And any kind of hardware that you put into a body is predisposed to getting biofilms, which are basically a slimy layer of cells and bacteria that are often impervious to antibiotics. So he'd had this staph infection for years and couldn't get rid of it, and his heart was failing and he was going to need a heart transplant. The doctor said, "We can't operate on you with this superbug." So he heard about Tom's case, came down to San Diego. We treated him as an outpatient through his PICC line. In 3 weeks, he'd cleared his infection, went on the heart transplant list, even agreed to take a hepatitis C infected heart because he said, well, I've injected legions of viruses into me. What's one more virus like hepatitis C when there's a treatment for that too? So he was out of the hospital before we could even meet him, and we met him for lunch. So it's just incredible, these stories of people that we've met whose lives and limbs have been saved through phage therapy as a result. And our chancellor at the University of California, San Diego, Pradeep Khosla, gave us seed money to start the first dedicated phage therapy center in North America called IPATH, or the Center for Innovative Phage Applications and Therapeutics. We just launched last summer, and Science Magazine posted an article the day that we opened. We have some funding promised by the National Institute of Health to begin a translational study of Pseudomonas aeruginosa in CF patients that will be beginning over the next several months. And we're really hoping to take phage therapy to clinical trials because we know that these single case reports are not enough to give data to the Food and Drug Administration to allow them to decide whether or not phage therapy could be licensed alongside antibiotics. So I'll close by one last case, which is really, I think, the future of phage therapy. What I didn't mention earlier when I talked about the life cycle of phage is that there's another potential life cycle as well. After the phage enter the bacterial cell, they don't always go through this phage rage where they multiply themselves right away. Sometimes they're lazy. They integrate their DNA into the bacterial cell DNA, and in the book we describe this as hitting the snooze button. We don't want this, first because it doesn't kill the bacterial cell. Second, because these phages that integrate into the DNA of the bacterial cell can carry toxin genes or antimicrobial resistance genes, which could potentially cause harm. And finally, they also can make the bacterial cell resistant to other phage. And so we don't want that at all, but sometimes for reasons that we don't totally understand, all we can find are these lazy phages, or they're called temperate phages. And in the case of this girl, again, this photo is shared with permission. This is Isabel Carnell Holdaway from just outside of London, England. And she has cystic fibrosis, had a double lung transplant, and had a systemic Mycobacterium abscessus infection. And this organism is a cousin to tuberculosis. And she had an open chest infection and nodules all over her body, and she was in hospice. She was so close to death. Her mother heard about Tom's case, approached the doctor. The doctor contacted us at IPATH, and Tom's doctor, Dr. Chip Schooley, who co-directs IPATH with me, we had heard about a researcher, Dr. Graham Hatfall at the University of Pittsburgh, who had a giant phage library of Mycobacteria phage that had been identified by students who were basically learning how phage and bacteria interact. So these phages had never been used therapeutically, but Chip Schooley said, Graham, would you see if any of your phages are a match for this girl? And he said, absolutely, I'll give it a whirl. All he could find, one lytic phage, which is the phage rage kind that we want, and that was discovered by a student in South Africa who isolated it from a rotting eggplant, if you will believe it. And two of the other phages were these lazy phages or temperate phages, which we don't want, but we do want a phage cocktails because, again, we don't want the bacteria to become resistant. So he was able to go in and cleave out the repressor gene that forced these two lazy phages to become lytic. So this became the first genetically engineered phage cocktail. The UK government gave permission, surprisingly, for this phage cocktail to be used because they said, well, technically it's not a GMO because you're taking away a gene as opposed to adding one. And Isabel received intravenous phage therapy based on the success of Tom's case, even though they were worried about it because, again, you're injecting foreign agents into somebody's body and technically it could kill you. But she was going to die anyway, just like Tom. And the family and the doctors decided to risk it all. And she left the hospital within a week. She responded so quickly. And so she's now learning to drive, working part-time, taking her A-levels at school, and we're in touch on Facebook. And she said, when are you coming to visit? I'll make cupcakes. Honestly, I never dreamt that the worst experience we could have ever had in our life would end up helping other people like this. So we decided that we needed to pay it forward and share our story so that others could learn not only about the threat of superbugs, but the potential for phage therapy to help resurrect this cure that has been around for over 100 years. But we obviously need more research to bring it forward. So our story is now a book. There's also a documentary and a major motion picture that are being planned. At this point, we want to thank all of you for listening and to thank the global village of people that have come together who were total strangers who stepped up to help me save my husband's life. And our view is that if total strangers can do this for one man, we certainly can do it for the planet. And my husband to say a few words.
I'll just say that what is unique about my experience was it was a parallel experience to what Steph has described. For me, a lot of the time I was in a coma for at least 3 months out of the 9 months I was in the hospital. And message to those of you who may encounter someone in a coma, I could hear while I was in a coma. So people would speak and I could understand them, though it wasn't like you and I are talking right now. And in the book, The Perfect Predator, you'll see interludes where I describe the hallucinations that I was having. And they're quite frightening and pretty amazing. The experience when Steph mentioned that I had squeezed her hand when she asked if I wanted to live, I was a snake. I was actually being filmed for a documentary about death. And so at that point, I had a problem when she asked me to squeeze her hand, and that was I didn't have hands. So I had to think about it, and I realized I could wrap my body around her hand and squeeze.
Yeah.
There's a crazy story for you. But I'll say, too, that my case is what I think of as evidence-based hope. It's growing evidence now with other cases, but my case was just a single N of one.
And with that, Tom and I are happy to take
Okay.
any questions. Jonathan has asked, are there inherent differences between phages and other viruses? Well, the main difference is that phages specifically attack bacteria, so they don't attack human cells, they don't attack other types of cells, and they obviously have to match to a specific bacteria. Some phages are very specific, like Tom's phage had to be matched to the specific bacterial isolate, or Tom's bacteria, not just the genus and the species, but the specific isolate. For something like MRSA, there are fixed phage cocktails that are available that cover about 80% of all of the circulating strains. So these phages have a broader host range. So it may be that in some cases we won't need to personalize phage for every single individual, which can be arduous. But imagine having a phage bank where all the phages are characterized and sequenced and purified and ready to go. In that case, you can find phages very quickly to match a bacterial infection within a couple of days.